Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal, taking you all to the next part of biomolecules. Now, as we have started the enzymes, we have to study the properties of enzymes and what are the different classes of it. So let's move on to the first topic, that is what are the properties of enzyme. Whenever I'm considering any enzyme, the enzyme acts as biocatalyst. You must be knowing that till now. So the enzyme act as biocatalyst. So the enzyme is nothing but here biocatalyst. So what does the catalyst do? I have told you in the last lecture that a catalyst is just speeding up the chemical reactions. So whatever the biological reactions are taking place inside a body or any organism, those reactions are basically speed up with their rate. So the rate of biological reactions biological reaction is increased. And how does the catalyst was increasing the rate of reaction? By reducing down the activation energy. Hope you are well versed with those topics. If not, go and check my videos on the introduction to enzymes. Got it? So let's move to what is next case of enzyme. Whenever I'm considering the enzyme, the role of enzyme is to speed up the reaction. So the enzyme speeds up the reaction. Now, what do you mean by speeding up the reaction? So in this particular case, whenever I am considering speeding up the reaction means formation of product is quicker. All right. So the product formation is quicker. So whenever I am forming the product at a faster rate with lesser time interval, then my enzyme is basically speeding up the rate of reactions. So in this particular case, the enzyme needs to bind to the substrate. Got it? So first of all, whatever the substrate is required for a reaction that must be converted into a product, that has to bind with the enzyme first. So enzyme needs to bind to a substrate which carries out a chemical reaction through which the product formation occurs. The product formation occurs. But do enzyme get involved and consumed during the reaction? The answer is no. Whenever the reaction is finished, the enzyme is isolated back into the system. So there is no enzyme is consumed during the chemical process. Got it? So there is no enzyme is consumed during a chemical process. These are some basic properties of enzyme. Moving on to the next or the main property of an enzyme, that is three-dimensional structure. Whenever I am considering the enzyme was made up of protein. Enzyme was made up of protein. Every enzyme in the system is made up of protein. And whenever I'm considering the protein has tendency to fold on its surface. Got it? Whenever I'm considering a protein chain, it starts folding, and then the particular three-dimensional structure is achieved. So whenever the enzyme is made up of protein, this is basically the folding of protein. The folding of protein has been occurred. occurred. So in this particular case, already the folding of protein has taken place and then the enzyme is formed. Now, why the three-dimensional structure is very, very important? That is one of the main thing where the structure of protein plays a crucial role. Whenever I'm considering the enzyme, in this particular case, due to due to folding, the stability of structure increases. This is my common point where I am increasing the stability by completely folding the protein. However, whenever I am considering any stability criteria, then due to the three-dimensional structure, there is still the formation of sockets inside those particular structure. So the folding and three-dimensional structure has sites for attachment. Now, there were the different criterias or the different molecules or moieties which were getting attached to the enzyme, out of which one was a substrate. Alright, so whenever the three-dimensional structure is considered, the 3D structure has substrate binding site as substrate binding site, which is also called as active site of an enzyme, In this particular case, this site helps substrate to bind to an enzyme. Whenever the enzyme has to bind to a substrate or substrate has to bind to an enzyme, that complete binding takes place through the substrate binding site present on three-dimensional structure of enzyme. The enzymes hydrophobic part is exposed in the internal side of three-dimensional structure. The three-dimensional structure is one of the important case where if there is any hydrophobic part present inside the enzyme, then that particular part has to be exposed inside the particular structure. Why? Because enzyme is continuously working inside the cytoplasm or matrix, basically the ground substances. So enzymes are basically works in ground substance. And the ground substance is comprised of high level of water molecules. So whenever the water molecules are exposed on the enzyme, then hydrophilic part has to be on the outer surface, while hydrophobic part has to be present on the internal side of the three-dimensional structure. So that's why the three-dimensional structure provides stability as well as it is providing sites for attachment to the different moieties of the enzyme. Got my point? Next one is optimum temperature. Whenever I am considering any enzyme, as it is made up of protein, the protein has its own temperature for denaturation. So, the enzyme should also possess the maximum temperature over which it will denature. What do you mean by denature? That means complete structure of an enzyme is disrupted. So whenever I am considering enzyme, right? So as the temperature is increased, as the temperature is increased, the activity of an enzyme will also increase. So the activity of an enzyme increases when the temperature is increased, but till a certain point, till a certain point. All the enzyme in an organisms are well developed in such a way that they are working at the optimum temperature or the temperature present within the body of an organism. If you are considering human, it is around 37 degrees Celsius. So whenever all the enzymes are working inside a body, they has to be working at their best on 37 degrees Celsius itself. So enzymes work with higher efficiency with higher efficiency when a certain specific temperature when a certain specific temperature is provided. Now, why is that so? Because on that particular temperature, the structural stability is highest. Got my point? So at that particular temperature, at that temperature, and that temperature, the structural stability of an enzyme is highest. That's why the speeding up of that particular reaction will be also at a higher rate. So whenever I'm considering the enzyme that is working at its highest efficiency, whenever I'm considering an enzyme that works on its highest efficiency at a particular temperature, such temperature is called as optimum temperature of an enzyme. Got it? So whenever the temperature at which the enzyme works at highest efficiency, at highest efficiency, that particular temperature is called as optimum temperature. Got it? So next case is optimum pH. Whenever I am considering the temperature, due to the excessive temperature, the structure was getting denatured. Denatured means breaking down. And why it was breaking down? Because of the breaking of peptide bonds. Got it? So whenever the temperature is increased, the peptide bond started to distort it. But whenever the optimum pH is the case, the pH is basically concentration of hydrogen ions. Got it? Whenever the pH is less, that is below 7, then the concentration of H plus are very, very high. And whenever the pH is more than 7, then the concentration of H plus is very, very low. Got it? So in the particular case of pH, are you remembering an amphoteric nature of amino acid? So similarly, as the enzymes are nothing but here proteins. So proteins are made up of amino acids, and each amino acid has its own nature called as amphoteric nature. It is nothing but here amphoteric nature. So whenever I am considering amphoteric, whenever the pH is less than 7, that means in the acidic condition, the particular protein or enzyme will act as a base. Alright, and what is the property of base? That is, they are going to accept H plus ions. So due to which, whenever the pH is changed, then the overall or net charges on the enzyme enzyme changes on the enzyme changes. So due to which the charge repulsion can take place. So in this particular case, the repulsive forces of similar charges can occur. Of similar charges can occur. So at one point, the enzyme will be neutral. The enzyme will be neutral and shows it maximum activity. So the pH at which the enzyme shows its maximum activity, such pH is called as my optimum pH for the enzyme. Whenever I am increasing the pH gradually, in that case, at one point, the enzyme will be at its maximum activity rate, and that particular pH will be called as optimum pH. Got my point, guys? So, whenever the gradual change in pH occurs, all right, charge density, the charge density on enzyme changes. At one point, the charge density is formed in such a way that enzyme shows maximum activity. Enzyme shows maximum activity, and that pH is called as my optimum pH. Got it? So this is nothing but a case of optimum pH. So now what is the particular case? Whenever I am considering the enzyme, here there is one image calling welcome enzyme school alumni. Poor Jerry, I told him working in non-ideal pH level would denature him. Working in non-ideal pH level will denature him. That is what happens. So whenever the enzyme works at non-ideal pH, the repulsive forces disturbs the complete three-dimensional structure of enzyme. Got my point? So let's move to the next part, that is type of substrate. So whenever I'm considering a type of substrate, what does that mean? The enzyme needs to bind to a substrate. The enzyme needs to bind to a substrate. The enzyme is need to bind to a substrate. That is my basic need of any particular chemical reaction. So whenever the substrate is binding inside the enzyme, it is binding, the substrate binds. The substrate binds to enzyme at active site. So whenever the substrate is binding to an enzyme, the substrate is binding inside the active site. So this active site, should have and set of or a set of amino acids, those can interact with substrate easy. So whenever I am having a set of amino acids, those can interact with substrate. Can I form ionic bond or covalent bond, guys? That should not be a case because enzyme is temporarily playing a role inside a chemical reaction. It is not a permanent part of the reaction. Got my point? Enzyme was not consuming inside a chemical reaction. So there should be, the interaction should be, the interactions should be temporary, till the reaction ends. The reaction ends by formation of product. By formation of product. Right? So the bonding should be taking place between enzyme and substrate till the reaction is completely done. So during that particular case, the number of hydrogen bonds formed between enzyme and substrate is going to decide that whether the substrate is specific for that enzyme or not. And for that particular case, many of the scientists came forward and gave us some of the models of enzyme substrate interaction that we will be covering in the next part. So now what are the classes of enzyme? So the classes are completely based on the functional aspect of enzyme. Got it? The classes are based on the functionality of enzyme. So what is exactly the function of enzyme? That is going to decide the class of enzyme. So now how does this function is going to be decided? That is based on the activity of enzyme and the type of substrate it is going to face. So the classes are based on activity of enzyme and type of substrate it is binding with. The type of substrate it is binding with. Got it? And the enzymes are very specific for their substrate. Because whenever the active site is designed inside an enzyme, the number of hydrogen bonds are basically very specific. And those number of hydrogen bonds are going to decide whether the substrate is specific for those enzymes or not. Alright, so this specificity is basically based on number of hydrogen bond formation. Got my point, guys? So now let's see what are the classes of enzyme. So there is going to be total 6 classes. As we have seen, OTH LIL, OTH little. Got it? So now how does these classes are segregated? Let's see according to their name. So the first class for this is nothing but my oxidoreductase. So the first class is nothing but my oxidoreductase. As the name suggests, there is going to be the oxidation as well as reduction taking place. Right? The reaction in which both oxidation and reduction occurs. And reduction occurs. So in that particular case, you are going to see oxidoreductase enzymes. So now for this, we can consider one example, that is alcohol plus NAD plus is basically giving me aldehyde and NADH. So alcohol to aldehyde is nothing but my oxidation process. So in this particular case, there is oxidation has taken place. But whenever I am considering NADH, this NADH is undergoing reduction to give me NADH. So NAD plus has undergone reduction. So this complete process is carried out by alcohol dehydrogenase. Alcohol dehydrogenase. So the enzyme is removing hydrogen from alcohol, that is basically converting alcohol into your aldehyde. That means it is carrying out the process of oxidation, and to balance that particular oxidation, the reduction is taking place. So why redox reaction is taking place? The redox reaction occurs. The redox reaction occurs to balance the energy levels of system. If it becomes imbalanced, then the complete cell might get disrupted. Got it? So that's why to balance the complete chemical energy inside a reaction, the redox has to occur. That means whenever the oxidation is taking place, simultaneously the reduction has to occur as well. Next case is of transferase. Now the name suggests it is going to transfer something. So the enzyme work to transfer the group. Now which group is it? Let's see. So if I am considering an example, that is nothing but glucose is forming glucose 6-phosphate. Glucose 6-phosphate under the presence of enzyme hexokinase. Are you remembering the enzyme? The kinase which is going to add phosphate. So their function to add a phosphate is basically they are removing one phosphate from ATP. Right? It is adenosine triphosphate, out of which one phosphate is removed, added on the glucose, and they convert ATP into ADP. So there is nothing but the transfer of phosphate group from ATP to glucose has been taken place. Got it? Next one is nothing but your hydrolysis. I have told you this before that if I'm considering hydrolysis, they use water. The enzyme uses water to break down the bond. So whenever the bond has to be broken down with the use of water, you just cannot add water and break down the bonds. So you have to add enzyme as well. So this enzyme uses water so that the bond can be broken down. So in this particular cases of hydrolysis, the example can be of different type, out of which one is your sucrose. Sucrose was broken down into glucose and fructose whenever I am using enzyme sucrase. That sucrase was basically adding up the water, and this water addition is basically broken down sucrose into glucose and fructose. Got it? Fourth case is lysis. lysis. So in this particular case, you don't have to add anything. You don't have to add anything. The enzyme breaks down the substrate. The enzyme breaks down the substrate. So without addition of water, whenever the enzyme is breaking down the substrate, those enzymes are belonging to the category of lysis. So in this particular, the example can be seen, that is histidine. Histidine is converted, oh my god. Histidine is converted into histamine. Histidine is converted to histamine by removal of one carbon dioxide. So basically one carbon has been removed out, and that is nothing but a breakdown of substrate. So in this particular case, one carbon dioxide is removed. That's why enzyme name is histidine. As the substrate name suggests, decarboxylase. Decarboxylase. So this enzyme decarboxylase, it removes one carbon from substrate. One carbon from substrate, and in most of the cases it is always a CO2. Next case is isomerases. Next case is isomerases. As the name suggests, guys, isomerases has to be playing a role inside the formation of isomer. So the enzyme that forms isomers of substrate, that is nothing but your isomerases. If I'm considering this particular case with an example, there will be your glucose 6-phosphate. Glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate, and for this the isomerase enzyme is basically used. In this particular case, as we have seen that glucose is belonging to aldoses category of carbohydrate, while fructose is belong to your ketosis category of carbohydrate. So these are nothing but my functional isomers. In this particular case, the functional group was aldehyde, while in case of fructose, it was here case of ketone as main functional group. Next case is basically here ligases. Alright. The ligases are basically used, or they are also called as synthetases. Alright. Ligases or synthetases. They are mostly used inside the biological processes of polymerization, DNA replication, where the gaps between the bonds, the gaps formed between two substrates to form a bond is completely filled. So ligases fills the gap between substrate by formation of bond, About this ligases category, we will be coming up with many of the examples in the case of molecular biology further. So that's why ligases is basically, for now, just remember that it fills the gap between substrate by forming a new bond. Got it, guys? So these are your classes of enzymes and what are the different properties. I hope you have loved this video. Please do not forget to like, share and subscribe to my channel, guys, and see you in the next video. Bye bye.